started with a new chapter which is in second year NCERT textbook, chapter number 2, sexual reproduction in flowering plants. So this is our third lecture in this chapter. In the first lecture we have seen introduction to the flower. In the second lecture we talked about the male reproductive organ, that is stamen, and stamen contains filament and anther lobes. Anther lobe contains microsporangium. The structure of microsporangium also we covered in the second lecture. Now in the third lecture we will talk about the dehiscence of the anther and how are the pollen grains released, and the structure of the pollen grain we are going to see in this lecture. So let us start today's class with dehiscence of anther. So here I have drawn a picture of a matured dehisced anther, right? So our today's topic is dehiscence of anther. Dehiscence of anther. Now we know that anther, they are the lobe-like structures which are present on the stamen. They are the fertile parts, right? And the anther, inside it contains microsporangium. This is one lobe of anther, this is the other lobe of anther, and inside it has two microsporangiums. Each lobe contains two microsporangiums, and we know that the anther wall has 4 layers. Outermost layer, this red colored layer, is epidermis. Then inner to epidermis is endothecium, hygroscopic layer, have callose thickenings. The green colored layer which I have drawn here represents endothecium. Now the third layer, which is in 1 to 5 layers made up of loosely packed parenchymatous cells, is called as middle layers. The brown which are represented is middle layers, and the fourth layer, which is a nutritive layer, is called as tapetum. So that tapetum will be consumed, which I couldn't draw here, it's not there, right? It's consumed by the sporogenous tissue. So this is one lobe of anther and this is another lobe of anther. Now anther lobe contains microsporangiums. Now here one microsporangium is there, here another microsporangium is there. Now the microsporangium inside, if the anther is young, it contains sporogenous tissue. When the anther matures, in the last lecture we discussed microsporogenesis. In microsporogenesis, we told that this is a sporogenous tissue inside a microsporangium, and all these cells are spore mother cells, or you can call them as pollen mother cells. These pollen mother cells will undergo meiotic division. The pollen mother cell undergoes meiotic division to make one pollen mother cell undergo meiotic division to 1, 2, 3, 4. What is this called? Microspore. Microspore tetrad we call, right? So the microspores, all the 4 microspores which are formed from one microspore mother cell are arranged together like a microspore tetrad. This is microspore tetrad. Now I think in the previous lecture I asked you a question like, find out for formation of 100 pollen grains, how many pollen mother cells are required? I asked you to post me in the comment box. Now you have seen here, from one pollen mother cell, if 4 microspores are formed, then if we have to get 100 such type of pollen grains, then I think 25 pollen mother cells are required. 25 pollen mother cells will produce 100, right, pollen grains or microspore tetrad, because one will make 4 microspores. If one is making 4, then if this is 25, 25 will make 100. So if you want to get 100 pollen grains, so 100 divided by 4, right, it will be 25. 25 pollen mother cells, if they undergo meiotic division, then we will get pollen grains. Is this your answer? Let me check in the comment box, right? So now we are talking that pollen grains are made in such manner. The sporogenous tissue, this is one microsporangium. Microsporangium contains spore mother cells. Spore mother cells are nothing but the pollen mother cells. The pollen mother cells have undergone meiotic division to make the pollen grains. Now these are the blue colored structures. What we are representing here are the pollen grains. Now the anther wall should split to release the pollen grains out. That's the concept called dehiscence of the anther. Okay, now what helps in splitting the anther walls means we have 3 types of dehiscence. One dehiscence is called as longitudinal dehiscence. Another dehiscence is called transverse dehiscence. Other dehiscence is called porous dehiscence. we have three types of dehiscence of the anther walls. One is longitudinal dehiscence, other one is transverse dehiscence, the third one is porous dehiscence. If you see NCERT textbook, in NCERT textbook, they showed us one anther. This is one anther lobe and here is the other anther lobe, and this is a filament they represented. And then they showed us a line here. And what is this line here? This is a line of dehiscence. What is this line representing? This is the line of dehiscence. Means if the anther lobes are breaking longitudinally, then that is called longitudinal dehiscence. And if you remember in China rose, in Malvaceae, we tell that the anther lobes are like this. One anther lobe will be there and it is dorsifixed. Now, if the anther is in such a manner, then it undergoes transverse dehiscence. Means the line of dehiscence will be like this. The line of dehiscence will be like this. What is this dehiscence called? This I told it is longitudinal, right? And what is this dehiscence called? Transverse dehiscence. And if you see members like cassia and all, there the anther lobes will be like this. Here is one anther lobe. This is another anther lobe. And this is a filament. So we can see it in cassia. And this I told we see it in hibiscus. And this longitudinal dehiscence is a common type of dehiscence. This is a common type of dehiscence. So in longitudinal dehiscence, the anther wall splits on the longitudinal section. In transverse dehiscence, the anther wall splits transversely. In porous dehiscence, can you see the pores here? The anther is having pores here. The pollen drains through the pores, they'll be released out. This is called porous dehiscence. Example is cassia. This is porous dehiscence. Example is cassia. Now, these are the three types of dehiscence. Now, let us talk about the factors which help in dehiscence. And we told the common type of dehiscence is longitudinal dehiscence. Now, what happens in longitudinal dehiscence? Let us discuss. As I told, this is a epidermis. What happens to epidermis at the time of dehiscence? At what happens to endothecium, we will see. At maturity, the epidermis. Okay, so we are telling at maturity, the epidermis shrivels. Means it shrinks. Right? At maturity, the epidermis, it shrivels, it contracts, it shrinks, it becomes dry. It shrinks and it becomes dry. So that's why very thin layer of epidermis we represented. It is dry, it is shrinking, it's shriveling, right? Now, next, what happens to the endodermis, endothecium? As we know that endothecium cells are hygroscopic. These endothecium cells at maturity, they become fibrous. Cellulose fibrous thickenings will get deposited on the radial walls also and on the inner walls also. So endothecium shows fibrous thickenings. The endothecium shows fibrous thickenings of cellulose. Where is that cellulose getting deposited, children? On the inner wall also and on the radial wall also. That is why we can also call endothecium as fibrous layer. Epidermis, the second layer we are calling endothecium. We can also call it as a fibrous layer because at maturity, the endothecium gets fibrous depositions of cellulose on the radial wall and the inner wall, and it becomes, it helps in dehiscence. This fibrous thickening only helps in splitting. It helps in splitting of the anther wall. What happens to the middle layers? The third layer is middle layers. The middle layer will support endothecium. The middle layer will support endothecium to split. They support endothecium. They help in splitting. Now, what happens to the tapetum? We told the tapetum, the inner layer, it is a nutritive tissue which is nourishing the spore mother cells. It is used up. Tapetum layer is used up. Now that means 1, 2, 3 are helping in dehiscence of the anther wall. They are undergoing fibrous thickenings. This is shriveling, this is shrinking. Endothecium is having fibrous depositions and it's becoming thick, fibrous. Middle layers will support this. Now all this will help in splitting of the anther wall. Now when all this is happening here at one place, the epidermis and endothecium, they don't show thickenings, and that region is called stomium. What is this region called? This region is called stomium. Stomium is a part of epidermis. Stomium is a part of epidermis. Now that at that stomium, what happens? The cells will remain as it is, thin and fleshy. The cells at the stomium will be thin and fleshy. Now there it splits. You can see here at the stomium, the anther wall is splitting. Why? Rest all it became dry. There it is moist and it is retaining the water content. There it splits. That is called the line of dehiscence. When this splits, then the pollen grains which are formed, we told like the spore mother cell undergoes meiotic division to make 4 microspores. It is called as microspore tetrad. Then the pollen grains will be released out. The pollen grains will be released out when the line of dehiscence happen, when it is splitting. Understood? So what we discussed in the starting of this class, dehiscence of the anther wall. There are 3 types of dehiscence. Longitudinal dehiscence, here is a picture. Transverse dehiscence, this is the picture. Porous dehiscence, this is a picture. Now in longitudinal dehiscence, all this happens. Whereas in porous dehiscence and transverse dehiscence, all this epidermis, endothecium thickenings will not happen. Local dehydration leads to dehiscence in porous and transverse dehiscence. Here and here we tell that all this is not applicable, only local dehydration. Only local dehydration leads to the dehiscence of the anther walls. Did you understand this concept? Take a screenshot, then we will continue. Right? Hope you understood till here. Now let us discuss about the microsporangium and the pollen grain ones. Oh. Right. So usually we tell that a stamen has 2 anther lobes. This is one lobe and this is another anther lobe. And each lobe contains 2 microsporangiums arranged parallelly all throughout the. They run longitudinally throughout the anther beetle, right? So this is one microsporangium, this is another microsporangium. At maturity, what happens, you know? At maturity, so the common walls of both the microsporangiums will fuse. At maturity, the common walls of this microsporangium as well as this microsporangium fuse. The common walls of the microsporangiums are fusing, so then it becomes a common chamber. It becomes a chamber like this. Let me show here also. At maturity, this is a common wall and this is a common wall. Here we have a common wall and here we have a common wall. They are fusing to become one compartment. They are fusing to become one compartment. And here stomium is there, right? Here stomium is there, here stomium is there. So now if you see, like there's only one compartment, one locule is there. At maturity, both the microsporangiums of each lobe, they fuse to make one cavity, to make one compartment. So it has only one locule. It has only one chamber. It is this chamber. Here also, it is this chamber. Clearly you can see it here. Now in one lobe, there's only one chamber. There are no separate 2 microsporangiums because they fused up. So when they fused up, what is there inside this? Pollen grains are there, isn't it? Now these pollen grains will come out when they open. Understood? So that's why we are not able to see that one microsporangium here, another microsporangium here, because they fused up. At maturity, this happens, and then now we are seeing that the pollen grains are getting released out to get pollinated. They will go and land on the stigma and they help in pollination. Now the things which are coming out are called as pollen grains. Now let us talk what is a pollen grain, isn't it? What is a pollen grain? So pollen grain is also called microspore, or we can tell the microspore only develops into pollen grain. So we know that spore mother cell is there. or pollen mother cell is there. Where is this spore mother cell or pollen mother cell? Inside the microsporangium. It has undergone meiotic division. This is diploid. It has undergone meiotic division to make 4 microspores. We call it as a microspore tetrad. We call it as a microspore tetrad, right? Now, it is surrounded by a callose wall. Now, the callose enzyme comes and splits. So, when this splits, now this is released out. Each microspore is released out. Each microspore is released out. This microspore is nothing but this pollen grain. Okay. So, we can call pollen grain as microspore also. And we can also call this microspore is haploid, right? Because after meiosis it is formed. When haploid stage is there, and that too it is a male one, we call it as a male gametophyte. The pollen grain represents the male gametophyte. The pollen grain is also called as male gametophyte. It is called as a male gametophyte. Gametophyte means haploid, and it's going to make male gametes. So, pollen grain is called male gametophyte, which is haploid. Now, what is the shape of a pollen grain? Generally, pollen grains are spherical in outline. You can see here, right? So, generally, pollen grains are spherical. They are spherical in outline. Now, what is the dimension of the pollen grain? The size of the pollen grain is 25 to 50 micrometers in diameter. So, what's the size? It is 25 to 50 micrometers in diameter. This is the dimension of the pollen grain. Understood? Now, what does the pollen grain has inside? The pollen grain has abundant cytoplasm, abundant reserve food materials, a prominent nucleus, right? So, what does the pollen grain has? A pollen grain has abundant cytoplasm, rich of nutrients. The pollen grain is rich of nutrients. Like what nutrients it has? Like starch it has, and it has unsaturated fatty acids, good fatty acids, right? So, it has unsaturated fatty acids. So, you can see here I draw the picture, I made it spherical, and I draw the dots here telling that it has abundant cytoplasm which is rich in the nutrients like starch and unsaturated fatty acids, okay, and prominent nucleus also. It has prominent nucleus. Now, the membrane which is surrounding this pollen grain is called as plasma membrane. We can tell that the cytoplasm is covered by plasma membrane. The cytoplasm is covered by plasma membrane. This is a plasma membrane then which is covering the cytoplasm. The pollen grain is further covered by spore coat, that we call it as a sporoderm. The wall of the pollen grain is called sporoderm. The wall of pollen grain, I am writing PG. The wall of pollen grain is called sporoderm. The wall of the pollen grain is called sporoderm. This sporoderm has outer wall and inner wall. So, now what is the outer wall called? What is the inner wall called? It has an outer wall and it is represented by inner wall also. Children, the outer wall of pollen grain is called exine. The outer wall is called as exine and the inner wall is called as intine. The inner wall is called as intine. Now, if we have to draw the picture in NCERT, they showed us, right? So, this is the plasma membrane. Surrounding the plasma membrane, it has another layer also. So, this is the other layer. What is this other layer which is inside? This is called intine. Now, it has exine also then. Now, this is the exine. This is exine, okay. This exine has ornamental designs. It is rough of course, and it has ornamental designs also. It is having ornamental designs also. And this is the pollen grain, which has its nucleus, which is having the abundant cytoplasm. It is having abundant cytoplasm, and it will have vacuole also, where the food materials are stored. It has vacuole also. And this is the outer wall. What is the outer wall called? The outer wall is called exine, and this is the inner wall. What is the inner wall called? Intine. And inner to that there is a membrane. What is this membrane called? Plasma membrane, which is enclosing the cytoplasm, right? So the outer wall I showed there, it is called exine. Inner wall is called intine. Outer wall, you can see here, you can notice here, it is rough, right? So outer wall is rough, but inner wall is smooth. The outer wall, which is rough here, is it continuous, or here outer wall is not there, here outer wall is not there, here outer wall is not there? So outer wall is discontinuous also, whereas inner wall is continuous only. The outer wall I made this much thick. Outer wall is thick, whereas the inner wall, one layer only I drawn. It is thin. The composition of outer wall is made up of a fatty substance called sporopollen. Outer wall is made of a fatty substance. What is it called? Sporopollen. The outer wall is made up of a fatty substance called sporopollen. Inner wall is made of, it is made of pectocellulose. The outer wall is made up of sporopollen, inner wall is made up of pectocellulose. Sporopollen is a highly resistant material of the plant. Sporopollen, we are telling, the sporopollen is highly resistant material. It is a highly resistant material. It's highly resistant material means it is resistant towards temperature, it's resistant to acid, base, and even enzymes also. It cannot easily undergo degradation by enzymes, nor high temperature, nor acid, nor alkali. That's why we are telling the sporopollen is a highly resistant material, right? So it's a highly resistant material. It is resistant to acid uridon, alkali uridon, temperature, and even enzymatic treatment also cannot degrade it. Okay, so we are talking about pollen grain. So pollen grain is formed from the pollen mother cell or the spore mother cell. After meiotic division, we get microspores like a tetrad, which is covered by a callose layer. When the callose layer dissolves, the microspore comes out, which only develops into pollen grain. So that's why pollen grain can also be called as microspore. And since it is formed after meiotic division, it represents the gametophytic generation. We call it as a male gametophyte. What is the shape of the pollen grain? It is spherical. It has different shapes, but generally it is spherical in outline. What is the dimension of the pollen grain, children? It is 25 to 50 micrometers in diameter. Now, what does it has inside? It has abundant cytoplasm, which is rich with nutrients like starch, and it contains unsaturated fatty acid. It has prominent nucleus, right? The cytoplasm is covered by a thin plasma membrane. And the wall of the pollen grain is called sporoderm. This sporoderm has an outer layer and inner layer. The outer layer is called exine. The inner layer is called intine. So exine, we can see there, it is very thick. It is rough also, and it is discontinuous. The place where exine is absent, it makes a germ pore there. This is a place where exine is absent. What it is forming? It is forming an aperture called germ pore. Whereas at germ pore, we can tell exine is absent, but intine is there. Through this germ pore only, the intine will protrude. It will come out as a bud, takes the nutrients, come out as a bud, and it's going to make the pollen tube. That is the significance of the germ pore. And then we are telling this exine is made up of a fatty substance, carotenoid substances, which is called as sporopollen. If you recollect our previous classes, there we told that the tapetum at the time of degeneration, it secretes Ubis granules, which contains carotenoids. Ubis is named after the scientist who discovered those granules, right? So these Ubis granules only contain sporopollen. They will only come and make this outer wall of the pollen grain, which is called exine. This sporopollen is highly resistant to high temperature, acid, alkali and enzyme treatment. Because of this only, pollen grains are stored as fossils, which we call it as microfossil. Because of this highly resistant sporopollen material only, pollen grains can be preserved as fossils, which is called as microfossil. Take a screenshot, we will continue. Right? So, understood this much? Let me erase it first. Yeah, we are talking about the spore wall, right? We are talking about the wall of the pollen grain and we are telling it is exine and intine. Now, the sporoderm is divided into the inner intine, right? And children, what is the outer layer? The outer is called exine. Intine is one layered only. That's why we told it is thin. Whereas exine is divided again into two layers called ectexine and endexine. So we'll divide now the exine into two more layers called ectexine and endexine. So we divide them into ectexine and endexine. Again, ectexine is divided into two layers, endexine is also divided into two layers. This ectexine is divided into two layers, endexine is also divided into two more layers. Now, what is the outer layer of ectexine? So the ectexine is divided into outer tectum and inner, we call it as inner endexine. Outer exine is divided into ectexine. 1 minute, I made a mistake. Yeah. So sporoderm is divided into outer exine and inner intine. Exine is divided into two layers again, ectexine and endexine. We will divide ectexine into two again. Ectexine is divided into two layers. Outer one is called sexine. Inner one is called nexine. This I went wrong. The ectexine is divided into two layers again. Outer one is called sexine, inner one is called nexine. This outer sexine is further divided into two layers and this inner nexine is also further divided into two layers. Now the outer layer of sexine is called as outer tectum. The outer layer of endexine is called as outer foot layer. The inner layer of this sexine is called as endosexine. Endosexine. And the inner layer of nexine is called as inner endonexine. This is called inner endonexine. So, if we have to see like from outside to inside, if you are going, the first layer outside if you see will be the tectum. Inner to tectum will be endosexine. Then inner to endosexine will be the foot layer. Then will be the inner endonexine. Then till here it is exine. Then comes the fifth layer which is only one layer. It is intine. So if we have to see, we are telling that this tectum is the outermost layer and this tectine only gives the ornamental designs, right? This tectine is responsible for ornamental designs of the pollen grain. So when we are drawing, so this is a outermost layer, right? So what is this outermost layer called? Tectum. Inner to tectum, it is endosexine. So let me draw that layer also. This is the second layer. This is the first layer and this is the second layer. What is it called? Endosexine. Then inner to endosexine, we told it is foot layer. Now this will change the color for better understanding. So this is foot layer. Little thicker. So it has depositions. So this is foot layer. Means it is the third layer. Now inner to foot layer will be endexine or inner endoexine. So that we'll draw it like this. So what is this called? This is the fourth layer called inner endonexine. Now inner to endonexine will be the intine layer, which is smooth, which is thin. So let us draw the intine like this. Now what is this? This is thin, smooth, the inner wall of the pollen grain. What is this called? Intine. This is the fifth layer. Now inner to intine, we told the cytoplasm of the pollen grain is covered by plasma membrane. So I need to draw that also. What is this? This is the plasma membrane. This is the plasma membrane, and inner to the plasma membrane, here is the nucleus and here is the cytoplasm. This is the cytoplasm. So this is the composition of the spore wall. The spore wall is called as sporoderm. It has outer exine and inner intine. So only this information is given in NCERT. If we have to little elaborate and see, exine is again divided into two layers. One is called ectexine, other one is called endexine. Endexine is same one layer, where ectexine divides into sexine and nexine. Sexine is further divided into two layers, nexine is further divided into two layers. The layers of sexine are tectum and endosexine. The layers of nexine are foot layer and endonexine. So that means altogether the exine is having 4 layers, whereas intine is having only 1 layer. And finally, who is that responsible for giving the ornamental design of the exine? Means it comes the tectum only. In which shapes, in which designs we have? Usually we have spiny. What I draw there is spiny tectine. Spiny tectum is one type of design, and another one we can draw it like this. Tubercules. So it has projections like this. If the pollen, and these are the germ pores, right? These are the germ pores. And what are these called? These are called tuberculous tectum. This is the second type that we told. Spiny, first type. This is tuberculous, and third one will be reticulate. In reticulate, the tectum will give such type, and these are the germ pores. Okay, so what is this? Don't get confused. So these are germ pores, and this is the outer design. What is this design called? Reticulate tectum. And sometimes even this exine also has smooth. Sometimes this also has smooth. So I can draw a smooth tectum also. This is a smooth tectum. Smooth exine is also seen, and this will be the germ pore. So we're talking about tectum. We are telling that tectum is the one which is responsible for giving ornamental designs for the pollen grain, pollen wall. The tectum can give spiny tectum. The tectum can make tuberculous tectum. The tectum can also make reticulate type of tectum. The tectum can be even smooth. So in this board, we discussed about the sporoderm and its composition. Take a screenshot. We will continue later. Right. Now let us talk about the deviations in the germ pore also. Hope you understood this composition of the sporoderm. Now, the place where exine is absent, there it makes an aperture, right? So we have seen here the place where exine is absent, it makes an aperture. That through that aperture only pollen tube will protrude and come out. So if the aperture is like this, then that is called as germ pore. So place where exine is absent, the place where exine is absent is called germ pore. Germ pore means if it is rounded. But sometimes, if you see any pollen grain, so it has a furrow type of opening also. Then that is called germinal furrow. So if it is rounded, we call it as a germ pore. The place where the exine is absent in the type of a furrow, then it is called as germinal furrow. It is germinal furrow when the opening is wider. When the opening is wider. I showed germ pore here. If it is rounded, then we call it as a germ pore. If it is longer and wider, we call it as a germinal furrow. Listen children, in monocots, germinal furrows are seen. In dicots, germ pores are seen. So germ pore, where do we find? Germ pore is found in dicots, whereas germinal furrows are found in monocots. Germinal furrows are found in monocots. And again, now how many germ pores are there? Depending upon the number of openings, we can call it as monocolpate, bicolpate, tricolpate also. So depending on the number of apertures, so we can call it as monocolpate pollen grain, C-O-L-P-A-T-E, monocolpate, bicolpate, tricolpate. In monocolpate, only one opening will be there. So it is common for monocots. Monocolpate means only one opening. This is common in monocot plants. And usually it will be like furrow only. Monocolpate means usually it has germinal furrow. Now, bicolpate condition means two openings. Means if you draw it, this is one opening, this is other opening. This is called bicolpate condition. Tricolpate condition, we draw it here. One opening, second opening, third opening. If three openings are there, tricolpate. Bicolpate means two openings. Tricolpate means three openings. Tricolpate means three openings, bicolpate means two openings, monocolpate means one opening. We told monocolpate is common for monocots. So bicolpate and tricolpate, these two conditions are seen in dicots. Bicolpate and tricolpate conditions are seen in dicots. Understood? So it can be two openings or three openings or one opening. But what is that opening meant for? Through the germ pore, the intine will protrude. This is the germ pore. Through the germ pore, the intine will protrude and it is making a pollen tube, right? It is making a pollen tube. So if it has three openings also, it is going to make one pollen tube only. If it is having one also, one pollen tube only. If it is having two openings also, then also it is going to make one pollen tube only. If the pollen grain, so this is a condition. What is this called? Pollen tube is formed through the germ pore. Now, in most of the cases, one pollen tube is formed. That's what I showed you. In most of the cases, from one pollen grain, one pollen tube only will be formed. Now, such a type of pollen tube formation is called monosiphonous pollen tube. What is such a type of pollen tube formation called? Monosiphonous pollen tube. But polysiphonous pollen tube formation, we can see it in Malvaceae, and see one pollen tube from here it will come. Another pollen tube from here it will go. Another pollen tube from here also it will go. So we can see polycolpate condition. Polycolpate condition. What do you mean by polycolpate condition? More than one pollen tube. Here you can see 1, 2, 3. This is rare. polycolpate condition is found in Malvaceae and Cucurbitaceae. Is found in Malvaceae and Cucurbitaceae. Now, what did we discuss in this board? We are talking about the pores, the germ apertures, right? If one opening is there, it will be long and it will be called as germinal furrow, characteristic of monocot. If it is rounded opening, we call it as a germ pore, characteristic of dicots. Depending upon number of the pores, if one is there, monocolpate. If two pores are there, bicolpate. If three are there, tricolpate. Monocolpate condition is characteristic again of monocot plants, grasses and all. Bicolpate, tricolpate is characteristics of dicot. Now, in most of the cases, even one or two or three germ pores, only one pollen tube will come out. Such a type of pollen grain is called as monosiphonous pollen tube or monosiphonous pollen grain. Whereas in Malvaceae and Cucurbitaceae, so how many pollen tubes are there? Means how many germ pores are there? From there, those many pollen tubes will be formed. What is that condition called? That is called polycolpate condition, seen in Malvaceae and Cucurbitaceae. In the next session also, we will discuss the remaining part of pollen grain. Hope the lecture is informative. If you like the content, like, share and subscribe to my channel. Thank you.